This video contains 40 practice histology questions for an introductory histology lab on the 4 tissue types: epithelial, connective, muscle, and nervous. Each question is timestamped below in the description. You'll have the first 10 seconds or so to determine the tissue type before I'll start commenting on how to determine it and give the answer. So if you need more time, you can pause the video. Below is a table of the 4 tissue categories with tissue subtypes that we'll be identifying. In most cases, we will see each possible answer twice. The complete list is in the description. Let's get started. So with this slide, we see a lot of different tubes or tubules. Here's one in cross section. Here's a longitudinal section. So we have a lumen in the middle here. There's a lumen right there. So from that, we can assume that this is the basement membrane that surrounds it, where our cells are anchored to. And since we have a free surface here, we're going to start thinking epithelial because it lines something. Overall, we have a single layer, and the cells are roughly cube or cuboidal shaped, so we have simple cuboidal epithelium. So we have a lot of cells on this video, and each one of these in the middle, they are empty. However, if these were living cells, they'd be full of adipose or fat that pushes or protrudes the nucleus towards the peripheral side. So they're spherical. When we cut through them, we're not always going to see the nucleus. So there's some of them over here where there's no nuclei. Overall, this is adipose connective tissue. It is a type of loose connective tissue. Here we see a mix of formed elements that are erythrocytes, red blood cells, white blood cells, leukocytes, and platelets, thrombocytes. All of those are suspended in a plasma, the extracellular matrix. This is a specialized type of connective tissue, blood. Identify the tissue from here to here. So that green line is roughly where our basement membrane is. Here is our apical surface, or where our lumen is, the hole here. Since we have layers of cells lining an empty space, we know this is going to be an epithelium of some sort. Because there's layers, it's stratified. Now, to name the type based on the shape, we look at the apical surface and we see these cells from here to here. They're a lot flatter and thinner. Therefore, we have stratified squamous epithelium. More specifically, this is non-keratinized because we can see the nuclei all the way to the apical surface. Identify the tissue over here. Now we can see this wavy looking appearance roughly going from left to right as it transitions right here into a different tissue type. The wavy appearance continues this direction. This is actually a junction between a muscle and a tendon. So right here we can see some striations or lines that run perpendicular to those fibers. This is actually a muscle tissue of some sort. Over here though, this is what we're interested in. We can see different nuclei all over the place with collagen fibers that are bundled together in roughly the same direction in a regular-like appearance. Therefore, we have dense, regular connective tissue. Now, in this slide, we see some cells, but primarily we see the extracellular matrix that predominates. We see these thinner looking fibers that are darker in appearance and these thicker fibers. The thinner ones are elastic, the thicker ones are collagen fibers, and each nuclei we can assume are fibroblasts which help maintain the extracellular matrix. This is areolar connective tissue. It is a type of loose connective tissue proper. There roughly is our basement membrane. You can see there's a little bit of tearing right here from preparation of the slide, so that normally these cells should be attached there. Up here is our apical surface that's bordering an empty space. In this case, it's actually a lumen. Now we can see that there's a single layer of cells. They're roughly rectangular in shape or columnar, therefore we have simple columnar epithelium. Now when you look at this, you might see the nuclei are a bit staggered and you might think, hey, is this pseudostratified ciliated columnar epithelium? And it's really not, because we can clearly see the cell borders, number one, but number two, that these little hair-like projections right there, those are actually microvilli, they're not cilia. They're a little bit shorter compared to cilia in that they're not quite as long relative to the length of the cells. Now, this sample right here comes from the lungs. Specifically, we're looking at alveoli or air sacs in the lungs where we have gas exchange. So because of that, we have empty spaces. So we're going to start thinking epithelium of some sort. If we look at overall the shape of these cells, they might be a little hard to discern, but there's a nucleus right there. Overall, here's the cytoplasm. It appears roughly flattened. You might make a case for cube shape, but they really are flat, like right here. Overall, we have simple squamous epithelium. So this sample comes from the thyroid, and we can see a lot of these circular-like structures. Now, in some cases, we can't see the borders between the cells, but other times it's a little bit more obvious, like over here, we can see the borders. Overall, we have a single layer of cells that are roughly cube shape, so we have simple cuboidal epithelium. Name this tissue from here to here. Here is our basement membrane. This is a sample from the skin. This is the dermis down here, whereas here is the epidermis. We see that there are multiple layers of cells bordering an empty space, so we're going to think epithelium of some sort. Because there's multiple cells, we're going to think stratified, and then we're going to name it based on the cell shape at the apical side. Overall, these cells are very flat, therefore we have stratified squamous epithelium. Specifically, this is keratinized because we do not see any nuclei here at the surface. Here we see a number of fibers that are roughly orientated left to right. We see those fibers, they do have some bifurcations or branches, so they don't completely go from left to right. Key thing here, though, is we see a lot of these striations or these lighter bands that run perpendicular to those fibers. But the key thing here is we see these darkened regions of those bands. Those are intercalated discs. Therefore, we have cardiac muscle tissue. So now we have cells with fibers that are orientated roughly left to right up here. But for this one, you really need to try and identify overall the shape of the cells. And to do so, we're just going to draw a few of these out. And you're not going to see the beginnings end just perfectly for these, but overall you can see that there's a nucleus, and on both ends the cell tapers down, so we have this spindle-like shape to our cells. We don't see any striations that are perpendicular to those, therefore we can tell this is smooth muscle tissue. Now another thing to note is that with smooth muscle tissue, it's involuntary, and oftentimes we'll see samples where we have longitudinal sections of the muscle tissue, and over here we have a cross section of that muscle tissue right there. So we have two different layers, but this is smooth muscle tissue. So with this, we can see that there's these clear empty spaces. Those spaces are the lacuna, and inside of that exists a chondrocyte. So therefore, we have a cartilage of some sort This one is hyaline cartilage, and it has this glassy-like appearance to it. Name this tissue from here to here. So there is our basement membrane. So we can see there are multiple layers of cells, so it's some sort of stratified epithelium because we have a lumen here or an empty space. We look at the shape of the cells at the apical surface right here, and for the most part, they do vary. It's oftentimes described as scalloped, but this is what's known as a transitional epithelium because these cells, they're able to change shape. So this is of the ureter. So if there is urine flow through there, this space opens up. These cells at the surface, they have to change their shape. We have transitional epithelium. So there's a few different things going on this slide, but what we want to identify is this circular-like structure right there. On one side, we have a basement membrane. On the other side, we have an empty space. So we want to look overall at the shape of these cells and how many layers they are. We have a single layer of cells, and overall they are roughly flat in shape. Here's another example over here, although the contents in the middle are missing. Here we have simple squamous epithelium. Now we've seen this one before in a previous question. We have a bunch of these dark staining nuclei that are maintaining an extracellular matrix. We can assume that those nuclei are going to be fibroblasts. The extracellular matrix is primarily comprised of this fluid as well as fibers. We can really make out the elastic fibers, these thinner, darker staining fibers, but the overlaying thicker collagen fibers that stain lighter pink aren't as easy to distinguish, and they're overlapping. Overall, we have areolar connective tissue proper, specifically a loose connective tissue. So again, we have a basal membrane, we have an apical surface over here. We note that there are multiple layers, so it's some sort of stratified epithelium. In this case, we name it based on the cells on the apical surface, which do vary quite a bit. This is called transitional epithelium because these cells are able to morph and mold into different shapes. This is a sample of the bladder. So if we have urine on this side and it causes that bladder or urinary bladder to expand, these cells have to be able to adjust and change shape in order to account for that expansion of fluid. Name the tissue from here to here. So there is our basement membrane. Over here we have a free surface. So we look and we see there are multiple layers of cells. So we're going to think epithelium of some sort. We name it based on the shape at the apical side. Overall, we can see nuclei here and they're overall flattened. Therefore, we have stratified squamous epithelium. Specifically, non-keratinized because we can see nuclei at the surface. Here we see a rather large structure known as a soma or cell body with a nucleus in the middle, and it has these axonal-like projections. We can see those projections running across all over the place. We see a lot of these smaller nuclei that vastly outnumber the single nucleus right here. Those are astrocytes. This is our neuron. Here we have nervous tissue. Basal membrane, free or apical surface. Because we have that empty space, we're going to think epithelium. It's lining something. We see there are multiple nuclei that appear stratified, but we can see some of these cells, such as this one right here, we can see it start from the basement membrane and extend all the way up to the apical surface. Therefore, it is falsely stratified or pseudo-stratified, And then we have the cilia right here, columnar epithelium. Now in this one we have a number of these cells where we can see the outer plasma membrane. In some cases we can see the nucleus that's been pushed towards the peripheral aspect of that cell. These are actually filled full of lipids, but it's lost during the preparation. Each one of these is an adipocyte. We have adipose connective tissue. In some cases, you won't see a nucleus because they're so large and we cut through them, we lose the nucleus. Name the tissue on the right. So this is a junction between a muscle and a tendon. On the left here, we have fibers going left to right with striations perpendicular to that. This is a skeletal muscle. The tendon, on the other hand, is going to be comprised of collagen fibers that are bundled together and are orientated roughly in the same direction, or this wavy-like pattern. Here we have dense, regular connective tissue. Now this tissue type is organized into these structural units called osteons. Inside of it, we have a central canal containing a nerve, artery, and vein normally. We have these darkened spaces, which are lacuna, in which osteocytes occupy. And these little channels between them are called the canaliculi. The outer ground substance is very strong, made out of hydroxyapatite. This is commonly known as bone, or also known as osseous connective tissue. Here we see fibers that are running roughly up to down. We see these striations perpendicular to those fibers, and we see numerous nuclei to those single fibers, which we do not see apparent beginnings or ends to those fibers. This is a longitudinal section. Over here we have more of a cross-sectional view where we see the nuclei on the outer part. We don't see the striations quite as well through the cross-sectional view. Perhaps over here in more of an oblique view, we can see those striations a bit more. Overall, this is skeletal muscle tissue. This sample comes from the tongue, and the tongue has muscle fibers and oriented in multiple different directions in order to function for both digestion and speech. So we primarily have an extracellular matrix with these dense staining nuclei. Those are going to be our fibroblasts. These pink bundles are in cross-section, longitudinal, they're oriented obliquely. There's really no rhyme or reason to their pattern. Therefore, we have dense, irregular connective tissue. So if you identified these bundles of collagen, that is these thicker, lighter purple staining fibers running left to right, and you thought that this was dense, regular connective tissue, you're mostly correct. In this case, though, we have a lot more elastic fibers, those darker, thinner purple staining fibers that are running that same direction roughly, that are overlapping our fibroblasts, which are those darker staining purple dots. This is a specialized type called elastic connective tissue, sometimes known as dense elastic connective tissue. It's very similar to dense regular connective tissue, but because it has that elasticity to it, it has that added benefit in addition to the tensile benefits of the collagen bundles. Identify this tissue here. Okay. Basement membrane, apical surface, free side, meaning we're going to have some sort of epithelium. We see a lot of layers, it's stratified. Look at the surface, these cells are flattened. We have stratified squamous keratinized, because there's no nuclei there, epithelium. So this is another example from the skin, and in this case we actually have some melanin pigmentation that's being secreted here at the basal layer from our melanocytes that gives us that darker skin color when we're exposed to sunlight. Identify this part down here. So very similar to the last slide. Up here we actually have the skin, so this is the epidermis, this being the dermis down below. What type of tissue is the dermis? Well, we see darker staining nuclei, but this is a trichrome stain, and it has a blue stain, particularly for the collagen fibers. So our collagen fibers we can see in cross-section here, a bit more longitudinal or oblique in different directions. Therefore, we have dense, irregular connective tissue. Same thing, just a different type of stain, gives us a different quality, able to see different types of structures. So if you thought dense, regular connective tissue, you're mostly correct in this case. This is just a different type of stain where we see the collagen fibers, but also we can see elastic fibers. In this case, we have elastic connective tissue. It's a type of dense connective tissue, but we see a good quantity of elastic and as well as collagen fibers to give us both the tensile strength and the elastic resistance to that tissue type. So here we see a number of cells, but more importantly, we're seeing an extracellular matrix, and that extracellular matrix is comprised of these blue fibers, which we've seen before, which are collagen fibers, but they're technically a specialized type, type 3 collagen fibers, also known as reticular fibers. This is reticular connective tissue. And those fibers, the spiderweb-like net mesh, helps keep all these cells held together in this organized format. So we have reticular connective tissue. So here we see some spaces, that's this white part right here, and inside it we have cells that live within them. The space is the lacuna, and the cell inside of it is a chondrocyte. Surrounding it is their extracellular matrix, it's a specialized type. This is a type of cartilage. This is elastic cartilage. Now we've seen hyaline cartilage, which just have this glassy-like appearance, but elastic cartilage has these elastic fibers staining darker purple in this case, that are interwoven between all of these different lacuna existing within the extracellular matrix. Another way to differentiate it is elastic cartilage has an increased number of lacuna and chondrocytes compared to hyaline cartilage, but this is elastic cartilage. So whenever we see these circular structures like this one, or this one, that clear space right there, that is our lacuna, and living within it is a chondrocyte. Now we can differentiate that from osteocyte because the outer network doesn't look bone-like. It has this gelatinous looking substance. Overall, this we can tell is a cartilage. More specifically, note how we have these elastic fibers that blend in and attach to our cartilage there. It can be difficult to differentiate dense regular connective tissue from our cartilage because they blend in together and they're very, very similar. Big thing about this in fibrocartilage, it is very strong both for compression and tensile resistance. So this one has a trichrome staining, so it looks a little bit unique, but overall we can see these long fibers running up and down. Perpendicular to that, we can see these striations. Note that the nuclei, they stain right here, and they are outside of the fibers. They might appear superimposed, but they are outside of them. We don't see clear beginnings or ends. Therefore, we have skeletal muscle tissue. Again, just look for the overall patterns regardless of the colors. Use that to aid you in determining the tissue type. So this is a special stain for Nissl bodies. Overall, here we have a soma. Inside of that, we have a nucleus with the dark stain nucleolus right there. The rough ER right here, those are our Nissl bodies. Overall, this is a neuron because we can see we have axonal processes out there. Surrounding it, we can see a bunch of these glial cells. that greatly outnumber our neurons. Therefore, we have nervous tissue. Again, regardless of the stain, just look for the overall patterns. Okay, we've seen this one before at 400 power, but this one is at 100 power magnification. Here we have an osteon. Inside of it, we have a central canal. Here we have an osteon with a central canal. Same thing, osteon, central canal. Haversian canal connects the two. Each and every one of these black dots in between, those are our lacuna, or the space, and living within them should be an osteocyte. If you look at overall the matrix surrounding it, it doesn't quite look cartilage. It has a hydroxyapatite background to it. This is bone or osseous connective tissue. Just note the overall structural differences. It's very similar to cartilage, but the end product is slightly different, giving us bone. This is just a special preparation of blood. Normally we see mostly our red blood cells, our erythrocytes that is, but we see a lot of thrombocytes, the platelets, and leukocytes, the white blood cells. We see those in a higher quantity because we've extracted that from a centrifuge sample of blood, thus the buffy coat where we see more than usual platelets and white blood cells. But this is blood. So here we see fibers running roughly left to right. Perpendicular to it, we can see some striation, so we're going to start thinking muscle of some sort, but that narrows it down to either cardiac or skeletal. And in this case, we see these darker plates. Those are interarticulated discs. Thus, we have cardiac muscle tissue. So this one's going to be a little bit more difficult, and it's going to be a process of elimination. We see a little bit of an orientation from left to right. So we're going to start thinking perhaps it's some sort of muscle, or perhaps it's a dense regular connective tissue of some sort. We note that there's no striations that are perpendicular, so we're going to eliminate cardiac and skeletal muscle. From there, we look and we see a bunch of these nuclei, and we think, okay, what might be outside of those nuclei? And we don't really see any distinct collagen fibers or elastic fibers, but what we do see is we can outline overall the shape of some of the cytoplasms to these cells. And we note that, hey, there's a bit of this spindle-like shape to it, where we have a nucleus in the middle and it tapers out on both sides. And you look around, you see that's pretty consistent all over the place. So we can deduce that this one is smooth muscle tissue. So there roughly is our basement membrane. We have a lumen here in the middle. This is coming from the stomach. Now, you could see there's individual nuclei here that border the bottom part along that basement membrane. You don't really see any nuclei above that, so we're going to think that this is a single layer, in which it is. The problem with this one is you can't really see the exact cell borders between them, but if we draw lines between the individual nuclei, we roughly get this elongated length right there, or it appears more column length. So we can tell that there's a single layer and they are column-like. So we have simple columnar epithelium. Because we have this lumen right here, it's lining something, we know it's an epithelium. So there's our basement membrane right here. We see a bunch of cells that are lining an empty space, so we're going to think some sort of epithelium. If you look below, you might see some connective tissues, some blood vessels, you see some hyaline cartilage here. That might help you determine where we're at. If you don't know anything about the trachea or the windpipe, you might be thinking or knowing what this is, but if you don't, we look and we see that there are multiple nuclei here that look like they're stratified. You can't really see the exact cell borders, but we see the cilia right here that's rather long compared to overall the length of the cells. From that, we can determine this is pseudostratified ciliated columnar epithelium. Again, if the picture isn't clear, you can always look around the rest of the slide and give you a good idea of where we're at. And if we know that, hey, this has to do with respiratory system of some sort, you might be able to determine that this is pseudostratified ciliated columnar epithelium. This will conclude our practice quiz on the 4 categories of tissues. Remember that the slides in your A&P labs will be unique, and to use this quiz and the 4 intro videos as a guide. I will continue to make more advanced histology videos in each system and will have them posted at a later date to help aid you in your human A&P histology lab experience going forward, so feel free to check those out.